couple of days before, we have a big snow, or we say blizzard. Yesterday, we delayed the cars for like a couple of hours. How are we going to do? Oh, well, you can take the snow out. What's the easiest way to do? You add salt there. Is that right? So you add salt into snow. What's going to happen? It's going to easily to melt it because it will be melting less than 0 degrees Celsius. So that means you bring the freezing temperature lower when you add substance. Sometimes, obvious example is salt. Okay, so how you understand that? In the snowing day, we add salt. They spray the salt. The snow is easy to melt it. The reason is the melting, or we can say freezing temperature is decreased. It's less than 0. Okay, that's what. Okay, this one you know, pH. pH everybody knows, but we just want to go over it, because that's a very important, actually a very important index. So what is pH? Negative log 10 of the concentration of hydrogen or proton concentration. The value is somewhere between 0 to 14. If it's at 7, we say it's neutral. Okay, what are the examples? Is a pH at 0? Battery acid. This is the reason. The battery not only recycled, you're going to put another bag and you label say it's battery because the acid's too corrosive. That's why they have to be treated separately as a trash. I would say it's a special trash because the pH is very low. The battery acid is almost 0. Now, how about the pH 1? This is a good example. It's stomach acid. So, bacteria will not grow easily in the stomach because of the stomach acid is very low, the pH 1. The only one can grow is the Helicobacter pylori because they can, using ureas, if they have a ureas, will break down urea, become ammonia, the ammonia will be buffering, the stomach acid go to neutral so they can survive. Is that right? How we mentioned. Okay, what is the pH is 14? That is approximately 1 mole sodium hydroxide. But how about the neutral pH 7? This is really about milk. Okay. Now here they say pure water has a pH 7. But I tell you one thing, it's not. We can do a measurement in the lab using pure water or distilled water. It's usually a little bit acid. I don't know why. It's like pH 5.5 to 6.8 or something. It's never pH 7. Okay? So this is easy. That's measured. You can easily understand the pH is measured, how acid in the solutions or in the environments. And that's a very easy index for the foods, and a very important index in the foods. So let's say the milk, the pH should be around 7. If the milk, when you measure the pH is already 4 or 5, that's not good, which means bacteria is already growing there. Okay, that's a very obvious reason. Now, we don't want a very low pH because it's kind of corrosive. And we don't like a very high pH sometimes. Although those are like sanitation. We call it a household ammonia, but it's also going to damage the skins or the human bodies in some way. So usually the neutral pH is good. That's why when we grow bacteria in the solution, it's always solution is neutral pH. And we have lots of the products to buffer pH of the solution. For example, PBS. For example, like Tim, when you're using the 0.1% buffered peptone water, that is a buffer to buffer the pH to the neutral level. Okay, so these are easy to understand. We just go over it. Okay, this is elements and small molecules in a cell, and you need to know 1% dry weight is in inorganic status. These are very important: sodium, potassium, magnesium, calcium, ferric ions, chloride, phosphate, sulfate. Those are important. And organic compounds right here: aldehyde, amino, carbon monoxide, hydro, keto, methyl, phosphate, and the sulfur hydro. You can see they contribute to almost all these important macro molecules in our body and in the food product also. So we will mention those. When we talk about the Maillard reaction, we're going to talk about the keto reacted with amino acid group of the amino acids, amino group of the amino acid. So we'll mention those later. This is a very good table. It just tells you all the structures of the name and their function. Okay, this is something, ATP, everybody knows. We just want to go over it, just do a review. What is ATP? Is adenosine triphosphate. Okay, what it looks like. I usually like to draw it. If you still remember it, sometimes I don't remember very well, but I do want to try. So you do this. Something like this. Sorry. About it. I want to try. So you do this. This. Here, we have this guy, and goes here, we have oxygen first, and this guy. Remember, the phosphate is always 5, it's a shared 5 electron. So this guy goes here, this guy goes here. And then we have, this is, they use an arrow here, that means it's important. Then goes here, we have another one. Okay, so what is this guy? This is called adenosine. What is this guy? Ribose. Okay, if we take one O, that means deoxyribose. Okay, what is this one called? That's called adenosine. Okay, where are the phosphates right here? Those are the phosphates. So, if this goes here, that means this is AMP. Goes here, that means ADP. If the last one goes from the top all the way, this is called ATP. Okay? Now, very important, we will mention later on. This guy, ADP except the phosphate becomes ATP. There is 2 functions can achieve this ATP. What is that called? Substrate level phosphorylation and oxidative phosphorylation. They rely on electron transport chain or without electron transport chain. Okay, we'll mention in the next lecture. And I always mention this AMP in my general microbiology class. This AMP, if they become ADP, ATP, that is good. How about if it's cycled, becomes CAMP? That's not good. Once they become CAMP, they cycled, which means like the money has been locked in your personal locker. You can't get the money. Because ATP is an energy currency for all the microorganisms to survive in the world. If this has been locked, you don't have energy currency, you're going to die. So all the bad things happen with this cycle. When you do, when you have a cough, sometimes the mucus generates related to this AMP becomes CMP. Okay, so that's just give you an example. So that's ATP, the energy currency of the cell. Now we're going to mention briefly about all these macromolecules and their component parts. So what means a macromolecule? Of course, it's a large molecule. The mass weight is higher or greater. There is 4 major classes. We know that: proteins, carbohydrates, nucleic acids, and the lipids. Now, in this class, we're not going to mention too much nucleic acids, because I already did that in general microbiology lecture, and also you have a genetic class talk about that. So we're not going to talk, after this class, we're not going to talk about that anymore. But we will talk in detail protein, carbohydrates, lipids. Those are three major things in detail for each section related to the food chemistry. Okay, we'll talk about that. Now, how these large molecules, the biosynthesis, or how they separate, it's very easy. If they attracting each other, combine each other, usually they will donate. They will be actually using warm water. So that's called dehydration. So they generate warm water. And how about if they separate? That's called hydrolysis. They will usually donate warm water, one molecule of the water. So they separate. So therefore, we had a very two simple terminology. Dehydration, you yield water. You contribute water. You donate water. Hydrolysis, you require adding the water. You use water. Okay, so that's very easy for all of them. Now, so first let's talk a little bit about the protein. You know what is protein? Protein is about 50% of the dry weight amount of a live cell that is composed by protein. And what's the function of the protein? You know, it's structure. It can be stress, stimulus, signal, gene expression, moving cells, transport molecules, and enzyme is also a protein. So it can be catalyzed reactions. So we all know these. You learned in the other classes, so we're not going to mention detail about here, but we want to just mention a little bit more. So protein, when we talk about the protein, they usually possibly the combination of 20 amino acids. And you want to make sure these amino acids is a possible combination. Now, let's do a math. How many gene code we need? Remember, 3 letters of the gene code translated become one amino acid. Each of them, each well could be A, T, G, C. This is like a very good math puzzle. So how many possibilities? 4 multiplied by 4 multiplied by 4 multiplied by 4 equals 64. By real life, how many gene code are responsible for 20 amino acids? Is actually 61. But where are those 3 comes? Those 3, or we say it's a nonsense code because it's a stop code. Okay? Just want to make sure you understand about here. So 20 amino acids. Amino acids, what are they looks like? What is this one? Carboxybase. That's negatively charged. What of this guy? Amino group. That's positively charged. Now, why all the amino acids are different? Depends on the R side chain decided different type of the amino acids. Because we have a carbon in the center, it's like a backbone. There you have 4 different chemicals attached to it. So if you put a mirror here, you got a mirror image, which is in the opposite direction. Okay, like you have a hands, you put a mirror there. This comes out a terminology called stereoisomers. Okay, now for stereoisomers, you need to know we have R, L, and R and D amino acid. These 20 amino acids composed by protein, they are all L amino acids. The D amino acid is very, very few in the real life. Some of the bacteria in an extreme condition, they may have a D amino acid. Okay, that's something we need to know. This is one you learn in your chemistry. You go over it really quick. So amino acid characterized by the side chain. Based on the side chain, we have a nonpolar, polar hydrophilic, uncharged, and the polar hydrophilic charged. That means acidic. What means basic? Basic, which means it's kind of like a hydroxy, OH. Okay, acidic is H. So this is acid, this you can understand it's like alkaline. Okay, based on that side chain. Now, when we talk about here, is when we talk about the mirror image L amino acid, D amino acid, there is one exception. Glycine on the top that do not have this characteristics. The reason is they have two H on the top and the bottom, so you're not going to feel that stereo isomerization impacts. Okay, so they do not have L and D amino acid. It all looks like the same. So exception is on the top is this glycine. GLY is exception. Okay. Okay, how they come together? How they come together? It's very simple. We already said all those macromolecules, they come together. All depends dehydration or hydrolysis. So what happens here? You're going to combine together right here. This guy going to be gone. Okay, so dehydration. Take out one water, then become water. This should be take out the ones, so the top, so the bottom. Okay. What is this going to be? Amino acid when they connected together all the way. Peptide. Is that right? Okay, and they can connect it with each other. Therefore, comes out a very famous four structure. Primary, secondary, tertiary, and quaternary. Okay, for the protein. What happened? Okay, when it's off, never mind. I can just keep doing this. It's a little bit challenge. I don't know why it's like that. Screen on. Okay, let's just keep doing that. You have slides. I can keep talking about it. Don't worry. I'll put the video on my YouTube channel so you can keep seeing the left one. Okay. I don't know why it happened. It's very weird. Let's talk about it here. So we have four different structures: primary, secondary, tertiary, and quaternary. So we want to talk about these one by one. Okay, let's talk about these. So primary. What means primary? It's easy to say. Sequence of amino acids. Okay, that's very simple. So we don't want to talk about too much. We already said dehydration, they connect together. Secondary. What are going to be the secondary looks like? The secondary, we have two good examples. Is beta pleated sheet, or we say beta sheet, and alpha helix. So that means the sequence of the amino acids, they are helical, or how we say it, spiral. Okay. If it's become spiral, it's become the beta pleated sheet. If it's helical, become alpha helix. That's second. Now, what is going to be the third looks like? This is something we can talk about. Tertiary structure. Tertiary structure. That is still the sequence of amino acids. But the sequence of the amino acids, because of the polar system, so it's going to be determined also by whether it is water soluble. So give you an example. If it's glue-bearing status, that is water soluble. If it's fibrous, okay, the good terminology should be fibrous, sorry. If it's fibrous, that is in water soluble or water insoluble. Okay, that is the tertiary. Now, how about the last one? The last one right here you can see is continental. This is peptide combined by multiple weak bonds. Okay? Okay, here is something we want to say. Now, next one, with the protein domain. We talk about the protein domain. The protein also we want to talk about is the protein domain, which means there is certain places protein add helix or add sheets is in a stable structure. And that stable structure is independent of the whole protein. Usually it has a specific biological function. That's a place is called the protein domain. Now in the slides we also talk about substituted protein. What is substituted protein? Substituted protein, which means there is a combination between protein and other chemicals. So for example, if it's with sugar, we can have glycoprotein. There will be sugar and protein. If it's lipids, we call it a lipoprotein. That is the lipids with protein. Okay? Now, protein could be denatured. If you use pH, higher temperature, extreme pH, that will be denatured. Some of the denaturing will be reversed, some will not be reversed. So an easy example, if you have an egg, you cooked it, it's denatured. But when you put it in the cold water, it's not going to be reformed. Is that right? Because that denaturing is not reversible. It's a very easy example. Okay, carbohydrates. Next one is carbohydrates. This one is a little bit, we're probably not going to draw, but I just mentioned briefly. Okay, carbohydrates. Carbohydrates. Well, we already talk about the carbohydrates. What is carbon? C. What is hydrates? So what is carbohydrates? This. The ratio between carbon, hydrogen, and oxygen is always like that. Why the carbohydrates is important? Because it is always an energy source. It is always a carbon source. And it is always important for the DNA, RNA structure, and it is always important for cell structure. Okay? That's something we want to mention. So you have a 5 carbon, 6 carbon. The next slide we talk about 5 carbon, 6 carbon. And the 5 carbon like ribose, deoxyribose. 6 carbon we have glucose, galactose, fructose, mannose. You can see them all on the slides. Have an example. Now one thing I want to mention. For the carbon, what happened is we have this guy. Okay? It depends on the position of these hydroxy bonds. If like that, alpha form. If this on the top, beta form. Okay, this on the top is beta form. Now, we're going to talk about disaccharides, which is we call it double sugar. Next slides. In the class, a little bit detailed. So sucrose, composed by glucose and the fructose. Lactose, composed by glucose and the galactose. And the maltose, composed by glucose and the glucose. Now, dehydration, you lose water, you add water, yields to monosaccharides. Okay, that's very easy and easy to understand it. Now, in this class, we also going to talk about detail of the polysaccharides. Polysaccharides usually is composed by oligosaccharides, and they are